Hi. So what we're going to do now is something called non-Mendelian genetics, which does not follow the laws of Mendel, and you will not get the expected ratios that we've been getting in step number 1, 2, and 3. So it's the genetics of any trait that does not fall into the simple single gene dominance that Mendel first described, and it does not follow all of Mendel's laws or expected experimental outcomes. And this is a picture of a calico cat, and the coloration is not inherited by in a Mendelian way. It's not dominant recessive. So all their coloration has to do with what's called a mosaic, where cells mutate as the coloration is developed through a process called inactivation. So if you want to learn more of the really cool genetics, which most of genetics is non-Mendelian, we have a course here that goes through more possibilities. So what we are going to go through in this class is something called intermediate or incomplete dominance, which you might think of as blending inheritance. Codominance, where you see two dominant phenotypes or traits show up. Multiple alleles, where there are not just two alleles like you've been seeing in Mendelian genetics, like big T, little t, but you actually have multiple possibilities. Sex-linked, where the gene is actually found on the sex chromosomes and therefore males and females get it differently. And polygenic inheritance, where there are multiple genes that are creating a characteristic. So this first one is called incomplete dominance or blending inheritance. And what you see here is a rooster that is gray instead of black or white. So neither allele is dominant. Alleles work together to create an intermediate phenotype, which would be the gray one. The parental phenotypes can reappear in the F2 generation. So you'll get those purebreds in step 1, like you did with the Mendelian experiment, except for now in step 2, the dominant is not going to show up like you've seen before. It's still going to be heterozygous, but now you're going to see an intermediate phenotype between the two parents. Now the ratios are going to change. Now your phenotypic ratio, which in step 2, they all looked alike. Step 3, if you remember, it was a 3-to-1 ratio phenotypically. Now you're actually going to get the genotypic ratio in terms of how it's showing itself. And so we will practice these problems so you understand that. And so an example of incomplete dominance, Andalusian chickens, that's what you see on the right. Snapdragons, I think the next picture shows a snapdragon. No, so the next picture actually shows the steps for the Andalusian chicken. And so you can see here this is set up in the 3 similar steps to a Mendelian experiment. You get the purebreds. That would be step number 1. So I want you to write step number 1 over by where it says P1. And then step number 2, you do get an F1 generation, except for now the F1 generation didn't come out as expected, where usually you'd expect black. So I want you to circle the black in that first line, but you don't get black. You get an intermediate, a blue, or some people call it gray. And then the third step here, you get all your variation, except for now you get more variation than you'd expect. So you get the parents reappearing, black and white, but then you also have this blue. And so phenotypically, I want you to write down the ratio, which is kind of written here, 1 to 2 to 1. Okay, so here are the snapdragons. Now, there's some problems with this diagram I got offline, and the first problem is they're using capital and small case letters, and that actually suggests dominance recessive. So let's see if this is dominance and recessive. So you get those purebreds in the first generation, which takes a while to get, and then you cross them. And so again, I want you to circle the one you'd expect to show up in the second step, or where it says the F1 generation. So go ahead and do that. But you'll notice that one does not show up. So what shows up is a pink one, which is intermediate. So you're still heterozygous in this step, but now you're not seeing the expected dominant. You're seeing the blending. And the last part shows a Punnett square. And so off to the side, I want you to write down the phenotypic ratio of this cross. And think about, is that different than what you saw in Mendelian genetics? So I want you to think about why it's not a good idea with these types of problems to use big R, little r. Why is that not a good idea? So go ahead and write down your answer. Okay, so now this is a different type of non-Mendelian genetics. It's called codominance. So neither gene is recessive or dominant. There is no blending, except for what your eye does, actually. Heterozygotes express both traits, and so these are called roan coloration, which you can see in cattle, too. And so the one in the middle kind of shows it the best. So you see your eye kind of blends it together, but if you go up close, you're actually going to see a white hair, and then you're going to see a reddish hair. So all of these are types of roan coloration. You may have seen it in horses. So AB blood type is a codominant situation, roan cattle, roan horses. These are all types of codominance. Here's what you're looking at for setting up steps. So red cattle, you're crossing it by white cattle. So that's so what step is that? And what would it have been in a Mendelian experiment to get that roan cattle? So I want you to write off the step to the left on that first line. So this is actually not set up step by step, but take a look. You take that red cattle, you cross it by white, you get a roan. What step is that? In the center, you're creating your purebreds, and then in the F1, you are getting that roan coloration. Now, what we're going to do in some of these problems to avoid the dominant recessive is we are going to use a base letter to represent a location on a particular chromosome, and so that we know this is the same gene we're talking about, except for now we're going to use two different capital letters to represent the two colorations. And so this big C at the base means it's on the same location, and the big R at the kind of exponent position tells us it is purebred. It has two big R's, and then the two big W's tell us that is white. And then so the F1 will be big C, R, big C, W. And again, so this is just to remind us we're not talking about two different characteristics. We're talking about one. And so that last one was codominance. The third one, multiple alleles, so more than two traits per characteristics. And so here you see all the possible blood types or alleles for the human population, which we talked about, A, B, O. In the middle are all the possible genotypes, and so you can be big A, big A. You could be AB, you could be AO, and likewise going down, these are all the possible genotypes. And so phenotypically, the blood types are what you see phenotypically. So over by blood type, I want you to write down phenotype. And then the alleles are showing the genotype. And so there are a lot of different blood types that you can have genotypically, but phenotypically, you can only be A, AB, B, or O. Sex-linked inheritance. This is a really cool one. And we are going to practice the pedigree for hemophilia because hemophilia is an example of this. I don't know if you remember with your flies. Do you remember which color eyes your fly had? So circle the color eye you remember dealing with when we bred flies way back at the beginning of the year. So now the gene for the trait is located on the X chromosome. The trait is expressed in males more often, as long as the male is the XY and the female is XX, which is not always the case in organisms. So remember, the male only has one X, and so he can only carry one gene. And so he has no way of masking if there is a problem gene. Like a female can mask it. So like if a female's mom has handed down a good gene, and the dad is handed down a bad gene, she can mask that problem. But males, because they only have one X, can't mask it. So the minute they get a bad gene, they are going to show a bad gene. So statistically, it's easier for a male to get a bad sex-linked trait because all you need is one bad gene. So recessive sex-linked traits are only expressed in females if they are homozygous. So it can happen in females, but the chance of the female getting it from two parents is just less likely. And females can be normal, yet be carriers of the recessive gene. And so I want you to write down for carrier, kind of off to the side here, heterozygous individual. And so white eyes is actually a sex-linked trait in Drosophila flies. So is hemophilia. Color blindness. Remember when I did that colorblind chart? So since doing that colorblind chart, I've had all males be colorblind except for one female. So a couple of years ago I had a female who was actually colorblind, so it can happen. Now this chart is possibly a little confusing because I don't have the box around the Punnett square. So I want you to look at where the gametes are in that Punnett square and where the offspring are. Just put a box separating the gametes from the offspring and then complete the 4 squares. So what this is showing, so again, so that we don't get confused with Mendelian genetics, dominant, recessive. I mean, and these are dominant recessive, but they're different than Mendelian in that the males and females are getting it different. So in Mendelian genetics, males and females got it all the same. And so again, we're going to use a base letter, X, X, and Y. And now that's going to represent this as non-Mendelian. And now we're going to use a capital and small case because you can have dominant and recessive situations in this. And so you can see here the parents on the top breeding. So don't get that middle X mixed up. And you put their possible gametes off to the side on the Punnett square. And you put them together, possible offspring. And so I want you to write off to the side, are there any females that are showing this trait, whatever it is? Let's say it's color blindness. Are there any female colorblind phenotypic people? So go ahead and write your answer off to the side. Here's a cross between flies, and so you can see here in the top layer right there, you're creating what are now the best you can do for homozygous individuals, except for now the fly, only male fly on the right with white eyes, only needs one bad gene, and so there's no way to get a second gene to be homozygous for males because they only have one X. And then all F1 offspring have red eyes, so that is similar to Mendelian genetics. And now in the last generation, you do get a 3-to-1. That's what you'd expect in Mendelian. But now you're seeing a difference with how males and females get it, and that is a non-Mendelian thing. And last, polygenic inheritance. Really cool. So when you were making your babies, I don't know if you remember, you had to wait for the letter A's to come in and the letter H's to come in. I think the A's were for skin color, and the H's were for hair color, right? That was very different than what you saw in Mendelian genetics, where you only needed two alleles, one dominant in the population, one recessive, to create the phenotypes that you saw. A lot of you have been asking about the situation of having babies if you are black and white. And so these are the offspring of this couple right here. And so depending on how the genes show up, you can have different color babies with polygenic inheritance. So there are multiple genes with additive effects. Height, by the way, is like this too, to influence the trait. So examples: height, skin color, crop yield. For plants, these would be polygenic inheritance, where there are multiple genes and multiple locations on the chromosomes. We are going to be practicing a problem set with this. You can use this handout. Hopefully you filled everything in. I will be checking this packet to see that you did all those things. Okay, great.